Welcome to Careers in Discovery, your window into the world of biotech leaders. On each episode, we interview the industry's leading executives to understand how they built their careers, what they've learned along the way, and the advice they'd give to their younger selves. Welcome back to Careers in Discovery. I am delighted to be here with Samir Ounzain of HAYA Therapeutics. Samir, welcome to the show.
Thank you, Tom, for the invitation. I'm a big listener of your podcast, so it's a great pleasure to be on it.
We're really pleased to have you. And I'm really interested to get into your story, Samir, because I think there's some fascinating stuff to talk about there. We always start with what are you up to now? And people may have heard of HAYA recently. Obviously, there's been a lot of press around some of the success you've had, which is fantastic. Congratulations.
Thank you.
But this is your life's work, really, isn't it? This is not just what you've been doing since you raised your Series A. This goes back much further. So tell us a bit about where it is today to start with, and then we'll dive into the rest of it.
Oh, so HAYA today is approaching this really important inflection point or proof point, which is translating a first in biology area of drug discovery and drug development into hopefully clinical impact. The company recently closed a Series A financing, which really has now fueled us to take our approach into patients and to really deliver on why we do this in the first place, which is can these new biological insights translate to meaningful, impactful medicines in areas where, to be frank, I think the industry and society in general is looking for safer, more accessible and more effective approaches. And that's really, we're at that point. We'll be transitioning into the clinic over the next 12 months. So it's really a critical moment in the company's history and the value proposition's history, which is translating from idea concept to potential clinical proof of concept.
Yeah, fantastic. And, there's been more and more attention on the dark genome concept recently. And we were introduced through your CBO, Rich, who talked with great excitement about the approach, and that's really what got him interested. So without giving any of the family secrets away, what's different about the way you're looking at this area of research?
So I think the way I would frame it is there's a huge interest now in the dark genome as a target space, but the way I always approach this was really trying to build backwards from the unmet patient need. So in particular disease areas. So HAYA really, our platform and our approach is how can we make common and chronic diseases associated with health span truly tractable and accessible to disease modifying therapies.
Right.
So if you look at the objective, the goal, our ultimate vision is enabling a world with an enriched health span. So how do you do that? Well, common and chronic diseases are really the major drivers and plaguing much of society. And if we look at those diseases, there's essentially three things that happen. People with those diseases feel really bad. The function, their viability, their vitality, their functions are severely impaired. And ultimately, unfortunately, there's really bad outcomes associated with it, be that survival or hospitalization.
Yes.
So really build back from those three principles. What you realize is these common and chronic diseases are at the tissue level controlled by how cells respond to the environment. So we really take a first principles approach, which is feel, function, survive endpoints, outcomes in patients. What is the underlying root cause, causal biology that leads to those feel, function, survive endpoints? And what we find is it's how cells are changing over time in response to the environment.
Right.
And what we've also learned over the last 20 years in the post-genome era is most of the common genetic variation that tunes your risk to these common and chronic diseases is in a region of the genome which actually controls how cells speak to the environment.
Yes, I see.
So using that as a fundamental principle that cells changing in response to environmental signals and common genetics are essentially what will trigger and drive the processes that ultimately result in these horrible outcomes. Let's reframe what we think about as biological target space and biological target hypothesis. Let's put the cell at the center. the cell is the fundamental unit of disease-driving processes. The cell is what speaks to the environment to drive disease. So then if you go the layer below that and say, well, what inside the cell transacts that information processing? This is what takes us to the dark genome, which is now becoming obviously an exciting area. So the dark genome is the 98% of your genome that does not code for proteins. It's not protein coding. And essentially what's really emerged over the last couple of decades, it's the information processing portion of the genome. It's how your genome speaks in a very context, temporal, spatial, and disease-specific manner to the environment to regulate cell states.
Yeah.
And it's also where all the human genetics is found that predisposes you to those diseases. So where does HAYA come in? Well, HAYA's really pioneered how do we understand the emergent causality from the dark genome that controls these cell states? And what are the molecules that transact that process? And it turns out it's RNA. So the biological molecule that is really the integrator or the conductor of this information processing in the dark genome, it's a whole new class of RNAs that are non-coding in nature that mediate these effects. So HAYA, we have a relatively simple story, which is we're building backwards from the unmet patient need. We've gone to the root cause biology, and leveraging genetic medicines, we've developed a platform and a toolbox that allows us to, using these RNAs, so in an RNA-guided fashion, precisely reprogram disease-driving cell states. And that's how we think about the dark genome. We really think about it coming back from the causality of what impacts the patient.
Yeah, that's interesting. And I think a lot of, there are more and more companies that are looking at the practical uses of, and as you know, we talk about the dark genome, but as you say, it's 98% of the genome. This isn't like a sort of one target area. This is a huge amount of ground to cover, right? So lots of space for everybody there. But I think a lot of it has come out of very foundational research, which perhaps we're still figuring out, but what's the link to the patient? Whereas you've looked at it from the other way around from the start, it sounds like.
Well, I think as I transitioned from academia to industry, I changed my framing around the problem.
Okay.
My background was really obsessed with trying to understand one simple question, which is humans have around 22,000 protein coding genes. We have about 22,000 genes. And most of our, neighboring animal kingdom actually have a similar number of protein coding genes, whether that's a fly or a worm or, a mouse. And this was really the intellectual shock of my life when I started my bachelor's degree in 2001. In molecular biology, the human genome was published when I started my bachelor's degree.
Okay, yeah.
And that was when there was this huge realization that at the level of genes, how genes were described then, we have the same number, complement, and functional diversity of genes as a fly. So I had one obsession, which is, well, why are we different to a fly?
Yes.
How can we from our brains and from our societies, we can create culture, music, science. Well, what is the origin of those processes? What in our genome? If you're a reductionist and believe you are your genetics, well, what about our genetics allows us to be so dynamically plastic to our environment and to essentially allow the emergence of culture and music and all these things? And that was what actually my obsession was, which immediately took me to the non-coding genome, because what you see when you compare humans to other, I don't like to use the word more simple organisms, but let's say organisms with less plasticity in terms of environmental, interactions and the emergence of some of these complex things that humans derive, was there's only one thing that actually seems to scale with that type of complexity, which is the content of your non-coding dark genome. The dark genome scales with evolutionary complexity in terms of cellular plasticity, cellular diversity, and all those emergent things that evolve from complex neurons and complex tissue.
Yes.
So that was my obsession, actually, for most of my career, was trying to understand what the dark genome does and how does the dark genome contribute to differences amongst the animal kingdom. But not surprisingly, I very early in my career was doing that work in a translational setting, because what we started to realize post the genome being sequenced was all this GWAS studies, these genome-wide association studies, were finding variation in the non-coding gene. And actually, that's where I did my PhD in the laboratory of Sir Nilesh Somani at the University of Leicester, Glenfield Hospital. And his laboratory and department was pioneering genome-wide association studies for common and chronic cardiovascular diseases.
Yeah.
And I remember at the time that some of the foundational papers were being published by the lab, and there was all these highly associated variants that were not mapping to protein. And so that was definitely part of the catalyst that gave me a translational vision as I moved forward in my career.
Yeah. It's interesting. The image that popped into my head as you were talking about that is obviously in a different context. I think it's David Sinclair talks about the piano and the piano player, right? The genes and the epigenetics. Yeah. And it's kind of like, yes, it's interesting to learn the piano, but it doesn't tell you what made Beethoven tick. It doesn't tell you what made Mozart special. It's that sort of idea, right?
I love it. And, I'm always there's always an ongoing discussion about a metaphor for the genome. Is it a blueprint? Is it an operating system? Is it software? I think it's none of the above and a bit of all. And I think one of the interesting kind of metaphors is thinking about the genome as a musical score. So there's all of these variables that influence how the score is perceived, whether it's the conductor, the tone of the instruments, the age of the violin, all of that manifests in terms of a orchestral piece. And think of the genome a little bit like I like the metaphor of the score, the musical score.
Yeah, that makes a lot of sense. And you talked about some of the timing elements that played into your career, and we'll talk about those. But just before we started recording, you told me that this is a journey you've been on since you were 10 years old, and that I didn't ask why, but that fascinated me. So please take us back to the start on this, Samir.
Yeah, I mean, what's amazing with this story is there are multiple scientists I've realized who had the same trigger and spark in terms of molecular biology. So I think it's really interesting how cinema can influence your whole life. Now, when I was 10 years old, 1993, I'm giving away my age, but 1993, my mum took me to the cinema in our little town, and it's a moment that absolutely transformed my whole life and my vision of what I wanted to do in life, which was going into the cinema and watching Jurassic Park.
Okay.
And literally that movie, from leaving the cinema, I knew I wanted to work on DNA. I didn't know the term molecular biology or what it meant, but from that movie onwards, I really became obsessed about understanding the molecular basis, not just of life, but how a thing that always I was really passionate about was how do we interact with our environment? I think most of the interesting things in biology at every layer are linked to how we interact with our environment, whether that's with each other, whether that's with technology, whether that's with the fellow animal kingdom. And those two things were very aligned, which was Jurassic Park sparked my interest in the genome. There's actually a code there that could potentially explain all of these complex, let's say, higher level things around biology and how we behave at a molecular level. So I guess I've always been a reductionist from a very young age, influenced by my father and family members who were also scientists. And then I realized, wow, the genome, this thing called DNA, could actually start explaining some really complex things. And that triggered my obsession actually with molecular biology, and ultimately my whole career was that movie.
Interesting. And I assume from what you've said, you took the moral of not trying to genetically engineer dinosaurs from the film as well.
Yeah, I mean, that was a nice, that was a nice kind of storyline, but it was really more around seeing these scientists playing with DNA and what you could do with DNA. And that was really, for me, just incredibly inspiring at that moment.
Hiring in biotech, especially in today's market, needs a certain level of clarity, and the difference between a good hire and the right hire isn't always clear. This podcast is brought to you by Singular Building Brilliant Biotechs, and we're offering listeners a free talent lens designed to show you where the best talent is, what they want, and how to win them. One hiring manager who used this recently told us, This is a really helpful breakout, especially the benchmarking aspect and the stratification of years of experience. I'm looking forward to a successful search. To get your free bespoke talent lens, email Kate, spelt CAIT, at singular-biotech.com now.
You mentioned just as you started your undergraduate, the human genome was sequenced. And I suppose, we'll have some people listening to this who were part of that project. We'll have some people who were scientists, professional scientists before that happened. We'll have some people who sequencing the human genome is just a thing that happens. Like, it's just that space that. So, like, going into, you'd had this obsession for a number of years. You're going in to do your undergraduate. You probably feel a little bit like, right, I'm going to be a proper scientist now. And this thing happens that is foundational to everything that's come after just at that point. Tell us a bit about, like, sort of if you can remember, the before and after of that. You said about this was one of the biggest realizations you had when you saw the output of that.
So, just the context, I was obviously going from high school. So, my understanding of molecular biology, especially in those days, molecular biology wasn't let's say, a highly covered topic in high school biology.
Right.
So it was really coming from, magazines like New Scientist and things like that. This was also when New Scientist was a great way to find out what was happening. So there was always, just to be clear, the molecular biologists, the genomicists, the scientists from the 80s onwards, already had a lot of clues and evidence that the genome was actually primarily non-coding. But I think what the draft sequence, because the draft sequence was published in 2001, obviously we had the full in 2003. I think that really crystallized this concept of coding versus non-coding, especially across species. And I think the realization was that now we had the full book of life, and at the level of genetics, from a protein coding centric view and a protein centric view of biology, I think that was a moment where a flag was really laid down, which challenged people in a different way. And for me, it was just incredibly exciting, actually, because now we had this first draft and, we had a pretty good understanding of just how many genes does a human have that is responsible for all of our complexity. And, I think at the time it was about 28,000. It's actually keeps shrinking. I think now we're at around 21,000. But going into the Human Genome Project, there was all these different opinions. Do we have 200,000? Do we have 2 million? And in the end, we landed at around 25,000.
Yeah.
And the mouse genome was published at a similar time, and they had the same number of genes. And we already had the worm genome and the fly genome. And I think it was just such an interesting moment in the history of molecular biology, and also both very junior, early stage scientists like me who was just starting my bachelor's degree. I was just starting to learn about biochemistry and molecular biology. But I felt like I was entering the post-genome world. And that was really exciting.
Yeah. And as time went on, as you said, you did your PhD at Leicester with a real sort of world leader in the field, which must have been extremely influential.
Yeah, no, it was. I was one of the only people in the lab that were not working on the genetics side.
Okay.
So my project was really about transcriptional control and gene regulation, because from bachelor's, I immediately had an interest in the non-coding genome. And at the time, there was already a lot of beautiful work done around gene regulation, And I think, at the time, it was very well understood that when you're thinking about gene function, it's not just sequence and structure of the protein, it's also regulation. The protein has to be expressed at the right place and at the right time in order to manifest its function. And so that was, how I was playing in the protein-centric view of biology, which is the sequence, the structure, which all drives the function, but all of that is meaningless unless the protein is expressed in the right place at the right time. And that is gene regulation, transcriptional. Yes. And that was my focus, especially these regulatory elements in the genome that control how mRNAs are transcribed at the right place at the right time. So that was my focus during my PhD.
That makes sense. And at what point did that sometimes it's hard to put your finger on these things, right? But at what point did that start to become the concept that HAYA has been built around?
So it's very weird. I've had 2 or 3 moments in my life where things crystallized. There was a moment when that whole concept crystallized very, very clearly, which was 2009.
Okay.
So I'd finished my PhD. I was doing postdoctoral work looking at transcription control and gene regulation in cardiovascular development. UCL at this point. I was down in London. And I was becoming really interested in how these distal regulatory elements are actually regulating gene expression at a level beyond proteins, beyond transcription factors, beyond epigenetic factors. And around that time, 2009 to 2011, we'd had, post the genome sequencing, this exponential acceleration in sequencing technologies.
Right.
And we started to move to a point where not only could we sequence the human genome, we could start to look genome-wide at all of the genetic switches, all of these regulatory elements. But we'd also reached a point where we could start looking for RNA expression across all of the genome. And what was starting to become very apparent around 2008 and 9 is that most of the genome is actually transcribed. So most of the RNA produced by your genome isn't messenger RNA or pre-messenger RNA. It's from all these regions that are, and just to remind you, it was still called junk DNA.
Right, yes.
And that was kind of a bit of a eureka moment in terms of connecting some dots for me, which was I'd spent the last 6 years working on regulatory elements and how a regulatory element controls a gene, etc. And then with this emerging data that RNA is coming from all over the genome, I really started to think, well, RNA might be, and there was published papers starting to suggest this, might be important for the activity of these elements in the genome.
Yes.
And then, well, RNA potentially is tractable to being targeted in a programmable, highly specific way, i.e. oligonucleotides. And I remember at that point thinking, if we can understand the network of these non-coding regulatory elements that are controlling genes and eventually, take it to the next level, cellular states, we can potentially intervene and drug those RNAs to have disease-modifying effects.
Yes.
And that was really an idea I'd started to formulate around 2009.
Yeah, interesting. And then HAYA was founded in 2019, is that right?
Yeah.
I'm always interested in this transition then from, especially when it's an academic idea and that's where a lot of these things start. There's a point at which it becomes, we need to start a company, right? And some of that's to do with progress, I'm sure, and more data and more certainty in the concept. But there's also a personal decision to make there, right?
Yep.
Am I going to be an academic? Am I going to start a company? Okay, how do I do, if I'm going to do that, how do I do that? What's the sort of, there's the whole decision for you in your career as well. So tell us a bit about how that evolved to the point where you said, okay, this is something I need to spin out and pursue, and this is something that we need to take in a different direction. We need to develop in a company.
Yeah, no, great question. So 2017 is essentially when I made this decision, and it was a really interesting period in my career because up until that point, my only focus was to become an independent investigator, have my own lab, and spend my life trying to study the regulatory genome on dark genome.
Yeah.
Okay. And we'd actually, 2017, the previous 6-7 years, I'd really been driving this area of research in cardiovascular disease, and we started to publish some really important papers. So the first examples of identifying these RNAs influencing cardiac development, cardiac disease, and there was really starting to become some tractability as you start thinking about, maybe translation. But still up to 2017, my plan was, and indeed at the time, the summer of 2017, I was actually offered a couple of tenure track positions to start my own lab. So I was really at this point, which is I continue this biology as an academic or not. And ultimately, I think what made the difference for me was, as I really started to think about the potential of this biology and what translating this biology could mean to to patients, especially in many of these common and chronic disease areas. I was at a point very naive and I didn't know anything about drug discovery and drug development. And I was so frustrated. I was thinking, we have all these insights into the biology of the non-coding genome and how all these genetic variations linked to these diseases are found here. And the biology is there. Why don't we have medicines? Why are we suffering? Why is my friends, my family, and the broader society dying? Diseases actually where I believed at the time, naively, we should be killing these diseases. We should have better medicines. And that was really a strong motivator, because then I stepped back and I thought, what do I need in order to be able to actually unlock the power of this biology to make those medicines that I think should be existing today? And really at the time I'm a big believer in crowdsourcing information. There's so much information in the public domain. Podcasts like this, YouTube content was transformational for me over the summer of 2017. Yes. I consumed every piece of information I could find on building a biotech and what biotech meant. So thank you to you and others for putting this content out there, because it really had an impact on me. And I came to this decision, which is I actually believe that the fastest, most effective way to truly unlock this biology for medicine, it's not through the academic path, it's through this other path. So it was really that was the driver of the decision. And then I really focused 100% on this.
Yeah, no, that makes sense. And the other part of your, so you've been running this business for 6.5 years now. And obviously if you sort of look at where you are today, you're running a company of 50 plus people, Switzerland and the US, just done a great Series A, going into the clinic. That's of course not what happened on day one. And I'm sure the thing I find fascinating about founders, especially when they stay with the journey through these evolutions, is that if you're doing a good job, you're constantly moving into areas where you don't have expertise, you don't know what to do, you've not done it before. So I'm really interested in, you consumed a lot of content, which is great. I think it gives you a great set of syntax, right? And it gives you a great vocabulary. And then you kind of sit down on day one and go, oh yeah, but what do I actually do first? And what do I do next? And what? So tell us a bit about that sort of transition to I guess, I think there's always a part where it's like thinking of yourself as a CEO is the first hurdle to overcome. And then where you went from there. And obviously it's been a progression from that day.
Yeah, so I mean, how it went in 2017, I also was very fortunate that my best friend also happened to be a great scientist with translational drug development experience around medicines and gene therapies, and was coming towards the end of his PhD here. And, we spent every day together after work. We were always together, and most of what we talk about would be science and, these type of topics.
Of course.
And there was also that piece of the puzzle, which I should absolutely emphasize, which is this process, this decision was also coupled to the fact that I had a co-founder who I believed together we could actually make it happen. So the co-founder piece, Daniel Blessing, my co-founder and CTO, we kind of went on this parallel journey in 2017, and both had a lot of opportunities, and both decided, let's forget about all these other opportunities. Let's do this. So we had the science. We had two founders that brought different skills to the table, one on the basic biology of discovery, more on drug development and translation. And we started to learn some of the syntax and the language that made us at least appear like we had a concept of what it meant to build a company.
Right.
Then what we actually did, we did two things. We entered a startup accelerator. There's one in Switzerland, which is originally from Boston, called Mass Challenge, which was actually fantastic for us because we started learning more about, what it means to actually build a company, a business plan, a pitch deck. And we were very fortunate to be one of the winners of that towards the end of 2017. And we got a little bit of money, $40,000. And what me and Daniel did with that is we essentially packed in our, roles. So we went all in. And then we went to the US and spent about 3 months starting in Boston, going across the whole US, all the way to San Diego, meeting as many KOLs, investors, and other stakeholders as possible to tell them about what we were doing. So we really did kind of a 3-month roadshow.
Yeah.
Trying to actually get people to listen, believe, and then potentially mentor or support us in that initial part of the journey. And I'm happy to say that at the end of that, we actually had some really important, experienced folks who said, you're super early. It's first in biology, but we think you might be onto something. And how can we help? Can we make an introduction? Do you have questions? Can we? And that became then critical, because having real experienced folks who are willing to give you some of their real-time insights, then allowed us to start really thinking about the value proposition.
Yeah, that's interesting. And it's very much sort of the methodology around building a technology company, right? Rather than building a biotech company necessarily. I think there's not enough emphasis on doing that research first, not the scientific research, but the market research, essentially.
Absolutely. And I'm really proud, actually, that both our lead investor of the seed round was met, the initial interaction, even though it took a long time before we got them really ready to go, was met in that sit. Our scientific advisory board, members of our clinical advisory board. We got real global pioneers willing to support us very early. And when you talk about timing, it's quite interesting, because some of the members of our advisory boards now are in exceptionally high demand because of where the biology and where the modality space is at.
Yes.
And where we started building those relationships, it was still very nascent. The biology and the field was very nascent. So we were very fortunate that we could build those relationships, which have been really important for us.
Yeah, absolutely. And how did that trip change your thinking about the business or maybe it didn't change, but how did it form the business? How did it form your thinking around the business? Sorry, is the question.
So, I mentioned up top this concept of building backwards from the unmet patient need, but it also informed us building backwards, not just from the patient need, but from the commercial need. And one of the things we talk about internally is we want to bring some sanity to drug development in common and chronic disease. And why sanity? Well, safe, accessible, needed, and effective medicines. Medicines. That's what we're trying to build. And I think that 3 months didn't just bring a lot of clarity and articulation around the patient, the unmet patient need. It also allowed us to start building backwards, thinking about the commercial and the market access needs. Like, what does a drug in these indications, dosed in this way, what does it have to look like to truly become tractable? So I think that 3 months really provided the vision from, you could even say, the level of the medicine and what the commercial opportunity would look like. It forced us actually to really think carefully about that early. In an area, by the way, which at the time was completely out of vogue. And maybe this is a little lesson I would give. Yeah. One thing we believe in HAYA is, if we take a first principles approach around building backwards from the need and the causality, and we can satisfy the criteria to translate, we should focus on that translational piece. We shouldn't get too distracted by all of the noise around which indications are venture backable and which are pharma interested in, because we raised the seed round targeted for the development of a first in biology antifibrotic for heart failure.
Okay.
Now, heart failure and cardiovascular disease in 2017 and 18 was not in vogue at all. So there was very little venture money going into precision cardiology or cardiovascular innovation. That's flipped 180 degrees now. I would argue over the next 1, 2, 3 years, you're going to see a huge amount of interest in investments in cardiovascular. So, you have to adapt to the market, but you should always, if you truly have a high impact disease modifying therapy that can achieve that same principles, you need to be thinking about where are we going, not where we are.
Yes.
And for me, common and chronic disease was very clearly an area that the industry needs to go back to because of the impact it's having on society. Aging populations, we need better interventions for many of these common chronic diseases. So we kept on that path.
it makes sense. And I think what you've done there is you've looked at it. So where I see a lot of biotech companies tripping up is that, and I understand why, because it's the funding is such a critical part of building a biotech company. It's so capital intensive, right, compared to other sectors. But they might think 2 rounds ahead, but they think in terms of, what do we need to do to get to the next round? What do we need to do to get to the next round? Whereas what you've said is, okay, well, this is the big picture vision. This is the long term of where we're trying to go. The first step of that is this. So we need to raise some money to do that, which gets us to this point, at which point we probably need to raise some more money, but we're still heading in that direction. So you're not being buffeted by those winds of what's trendy and what's not. And it might make it more or less difficult to raise money given the market, right? But I think, especially experienced investors, they will see the difference there and be able to see, okay, well, actually, yeah, look, these guys do have a plan. They have a direction. Things might change, but there's a longer term picture that we're painting here, and we know where we're going.
I think so, Tom. And the areas where we're focused on, the underlying value proposition, this is common and chronic disease where we genuinely don't have a lot of options. So, I think the market will sort itself out. If you can truly, in an accessible way, impact those areas, there's going to be a return on investment. And that's how I've always thought about it, actually. There's no point unlocking this new area of biology that specifically does a certain thing, which is allow cells to speak to the environment, and not translate it in the disease areas where cells speaking to the environment is the major driver.
Yes.
So we tend to say we're building backwards from patient need and forwards with biological truth. That's how we think.
Yeah, I like that. And then I'm sure there's lots of things, but over 6 years, but what else are kind of those key lessons that you've learned? And I guess the way I like to think about this is, this might be hard to conceptualize because this is your life's work, but let's say someone gave you $1 billion today and you sold the company and everything was good outcome, all that kind of stuff. I'm not saying that's what you're planning to do, but let's just hypothetically say that happened. But you thought, I'm going to start another company.
Yeah.
What would you do differently second time around, do you think?
So I think what I would do differently is I would double down even more than we did on team.
Okay.
And by team, I mean the timing of when we augmented in certain areas. So we were really science heavy, science focused. I think potentially bringing in some more translational and development expertise at different levels earlier, I would do that. Because I think the impact that's already had for the organization, as we're thinking about, the unmet patient need and translating, it's very, very clear. And I'm actually more strong believer now that it's never too early to have that input. And I would even argue that, and it's a real-time lesson today, that even bringing in commercialization perspective very, very early. So I think that is definitely something that I would probably have done a little bit earlier in the cycle of where we are. Because I think you need to have a laser clarity and understanding of how this is actually going to have a commercially viable impact on patients as soon as possible. And it's a lot of work. And a lot of that stuff, I talked about consuming content. A lot of that stuff is not easy to find.
No.
Right? And it's really not. And that's where you get that through people being there and doing that.
And that's it. It's like, even the stuff that you can find, well, there's people who already know how to do this. So is it worth you spending the hundreds of hours to figure it out? It's there's always a bit of a combination when you're in the early stages, right? Because you have limited resources, and that's a thing. What would you do the same? What do you think? And I guess the way I think about this is, I think the long-term vision is clearly one of the things that's made HAYA successful so far. What do you think of the other elements? What are the things that you would keep if you did this again?
I think the culture that we've built within the organization has definitely been a key enabler. So it's an intense, high expectation culture, but I think it's aligned around what we're trying to do. And I think one of the things that has really allowed us to attract talent at every level within the organization is we're really trailblazing in biology and science. We're really unlocking a whole new way of thinking about drug development and drug discovery.
Yeah.
This isn't incremental. This isn't improving incrementally, delivery or, the 20th iteration of a modality to target the same target. Our vision is actually, let's see how we think about drug discovery and development from a completely different lens. Let's put the cell at the center and let's put the genome as the source code to do that. This is very, very motivating for high-performing, very smart scientists and drug developers and translational biologists. And I think that's what I would do the same.
Yeah, no, absolutely. And I think it's about having that clarity, isn't it? There's no right or wrong. Well, there's maybe a wrong culture, but there's lots of ways you can build a company. But it's about which one are you building, and that's going to appeal to some people, and it's not going to appeal to others, and that's okay.
Couldn't agree more. I mean, HAYA's culture won't be for everybody. Absolutely not. But our culture is to deliver on our vision. We have a pretty big objective, but we want to get there as fast as possible. Because genuinely, I'm getting older, and one of the things I'm learning as I'm getting older is how much more I'm having to experience loss.
Right.
Friends, family, and a lot of the loss. I mean, I've had really impactful loss recently in my life, and it's for diseases that are common and chronic. Diseases that we should be able to, with our understanding of the biology, we should be coming up with translational solutions. But I think we have to change how we're thinking about these diseases. I think we have to change the lens by which we're approaching them. And that's what we're trying to do at HAYA, not just for HAYA, but for the industry.
Yes.
We want to trailblaze and demonstrate that framing drug discovery in this way for these diseases can change how we think about common and chronic disease.
Yeah.
And that's a big challenge, and it's an important mission. And the culture we have, it's the question I would always ask is what are we doing can enable that?
I completely agree. And I think this is probably too big a conversation for us to get into in the time that we have left. But what you've also said there, which I think is very timely, is that, look, we are at a point in the story of the biotech sector where the way things have been done no longer works effectively, right? I think people are still clinging on to that old model, but I heard a podcast recently, the person saying this perhaps did have a bit of agenda, so the numbers may be a bit sort of massaged, but over the last 20 years, the return on investment in the biotech industry is about 4%. So there is a positive return, but it's about what you get from a bond, overall, aggregate, right? That business model doesn't work for the amount of capital and the amount of risk, right? So there is this need to rethink, well, how are we building these companies? Not the science. The science is better than it's ever been. The science is incredible. But how are we building the companies around it? How are we taking these things from concepts to actual drugs or diagnostics or tools? And how are we making that commercially viable? I think the way it's been done isn't sustainable. And so there is this challenge for all biotech companies now, I think, not only to do new things scientifically, but to find a new path in terms of business model, in terms of commercial structure, in terms of reimbursement is going to be part of it, right? And as we get into prevention and things like that, there's a whole bunch of topics there. It's a fascinating set of challenges.
I couldn't agree more, Tom, and obviously I am biased, but I believe that, what we and others are building can, bring some interesting solution to those problems, whether it's, we're using a relatively de-risked off-the-shelf modality, oligonucleotides. We know these types of medicines can be produced at scale. The cost of goods mean they could translate into, drugs that could treat millions of people. And the focus, this is the message I keep emphasizing, is biology first. Because what I is an aggregation around very de-risked targets, and you're getting a scenario where a lot of capital and a lot of brain power and human resource is all aggregating around the same set of 50 targets,
right,
with a slightly incremental improved approach for those targets. And most of those targets, by the way, are not going to be disease modifying in the diseases that are truly plaguing all of society. So I think there needs to be a little bit more risk appetite around first in biology approaches.
Yes.
And ultimately that's going to be driven by success. And, that's our focus. If we can translate this into impactful outcomes in patients, it will increase the appetite for these type of approaches.
Absolutely. Well, we wish you the best of luck with it, Samir. I think I think you're thinking about it in the right way. I think that the market is rewarding that at the moment, and the progress is there. Obviously, it's an important inflection point, as you said right at the beginning. I've got my fingers crossed for you.
Thank you.
Thanks for joining us.
It's our laser focus.
Thanks for listening. If you enjoyed the episode, please like and subscribe and share it with someone else you think will benefit. And for more podcasts, videos, blogs and insights, come follow me, Tom Froggatt, on LinkedIn, YouTube, Instagram, Medium or TikTok. See you next time.